I'm going to tell you a few stories today that of science that came out of the lab and made its way into biotech with the intention of treating patients for a number of different maladies. So the first story, it's funny. I'm a stem cell biologist trained in Irv Weissman's lab in blood forming stem cells, and I started my lab in 2007. And I was right around the, shortly after Shinya Yamanaka had published his seminal paper on reprogramming adult somatic cells back to pluripotency with these, what are now known as the Yamanaka factors, these 4 transcription factors. And I, like everybody else in the stem cell community, was pretty inspired by this work. So when I started my lab in 2007, a year later, and I was actually at Shinya's talk at the ISSCR conference in Toronto when he first presented his data, and it was just, mind-blowing in its elegance and potential. So when I started my lab, we thought about a little side project. So you may recall that when Yamanaka first did reprogramming, he used retroviral vectors to deliver these transcription factors. And if we were ever to imagine the full therapeutic potential, so the near-term therapeutic potential was disease modeling in a dish, which probably the retroviruses wouldn't have a big impact upon, which retroviruses integrate the genome, of course. But if we were to think about, the generation of autologous cells for cell and tissue-based therapies, we would probably wish to get those genomes full of retroviruses out of the equation before imagining transplanting such cells. So we and others started to think about this, and there was many approaches to try to sort of achieve Yamanaka's objective using non-DNA integrating methods. So cell-penetrating peptides, for example, was one study that got published, and other approaches. But we had a very simple approach. We know that, I was trained as a molecular biologist. So I think everybody here in the room knows that, DNA is the code of life, but it's a relatively passive molecule. And in order for it to do its business, it has to make its effector molecules, which are proteins. But there's this obligate intermediate of messenger RNA that takes the code from the DNA, basically in a slightly altered nucleic acid, out of the nucleus into the cytoplasm to the ribosome for protein synthesis. So we thought, well, why don't we just use mRNAs to achieve this? It's a non-integrating nucleic acid. It also had the bonus of, being relatively labile molecules. So, we wish to express the Yamanaka factors for a short period of time, but then not express them anymore once we had reprogrammed the cells. So it seemed like a good idea. But wasn't it strange that, RNA had been discovered to be the intermediate molecule between DNA protein synthesis in 1961. And here we were in 2007, and there was not a lot of people that were, in fact, almost nobody, that was using mRNA as a sort of a delivery vehicle for protein production. So people were transfecting DNA, people were putting proteins into cells, but at best, people were mRNA profiling for transcriptomes, but nobody was using it as a tool. And molecular biologists use anything out of nature as a tool if they possibly could. So it was a little bit odd that it wasn't being used, but we sort of ignored that and thought, well, let's give it a try anyhow. So we turned to the textbooks, non-mRNA biologists per se. So we made very, canonical mRNAs. And so there's an open reading frame, 5' UTRs, 3' UTRs, a cap, a poly-A tail. And we transfected it into human cells in addition. It totally didn't work. And it didn't work in spectacular fashion. So not only did we not get, transfection and expression of the proteins that we so wished, and in the early days, we were just making a GFP mRNA, green fluorescent protein, but we were actually killing all the cells pretty robustly. And the reason for that is this rather busy slide. It turns out we did some profiling the cells before they died to find out that they had this massive NF-kappa B mediated interferon signal that was blasting in them just before death. So pretty much if you introduce a nucleic acid from outside the cell, so here at the top of the slide is the outside of the cell. This is the cytoplasm here, this large sort of beige area, nucleus down here. If you bring a nucleic acid in from the outside of the cell, cells have basically see that as bad news. That's by and large a pathogen coming in, be it a single-stranded nucleic acid or a double-stranded nucleic acid. And ever since, pathogens and cells first met one another, pathogens have been trying to figure out new and improved ways to infect cells, and cells have been coming up with new and improved pathways to detect and shut that down. So TLRs and NF-kappa B mediated mechanisms, NDR, RIG-I, and like I said, many others molecular players that are depicted in this slide. And so what we were doing when we were transfecting cells, we were tripping these pathways. We found out by profiling, which of course led to the cessation of protein translation, which is of course exactly what we wish to have happen, and apoptotic cell death, so programmed cell death. So better for a cell to be altruistic and kill itself upon viral transfection than to allow the pathogen to propagate and compromise the whole organism. So the cells were doing a good thing by killing themselves, but this was not going to really help us get over get to the goal that we wished, which was making these induced pluripotent stem cells. And that could have been the end of the story, but luckily there was a set of researchers out there that were sort of looking at this carefully. So as it turns out, the nucleosides of RNA are actually highly modified in cells of the body, in mammalian cells. So there's over 110 different modified nucleosides that are in use in the RNA in our cells. And really critically, in 2005, Ketelin Kariko and Drew Weissman at UPenn published a paper where they discovered, and I'm sure you all know this now because it's been widely celebrated and for good reason, they showed that certain modified nucleosides could be attached to RNAs. They weren't actually making mRNAs at the time. They didn't make any mRNAs in 2005 in their paper, but they showed that if you made oligonucleotides or longer oligos of RNA, that if you incorporated these modified nucleosides, which were naturally occurring, that you could abrogate this antiviral response. So we thought, well, maybe these modified nucleosides could be incorporated into mRNAs when we were synthesizing them, which is easy to do. When you synthesize mRNA, you have a cocktail of nucleosides, and we could just swap out one or more modified nucleosides. So here's an early experiment that we did where we transfected again a GFP construct, mRNA construct into cells. This is very low mag. I don't have a scale here, but these little green dots, which I hope you can see in whatever light you have in the room there, are indeed green cells. We got a few green cells, but we got a big plate of darkness and black here. And if you look over onto the right, you see actually that the viability on this lower graph on the right, the viability of this transfection is but 20% of the cells were positive, and that's upon a single transfection. We got low level. The graph above is the mean fluorescence intensity, so it was low level. So that's just using standard nucleosides. If we, however, swapped out a modified nucleoside, 5-methylcytosine, in place of cytosine, which Drew and Kathy had shown was decent at abrogating this antiviral response, we now got much greater viability down here on the lower right. You can see a lot more green cells. You can see that they're brighter. The mean fluorescence intensity was higher. Similarly, if we made mRNAs with pseudouridine in place of uridine, again, it had a good impact on cell viability and mean fluorescence intensity, so more GFP was expressed. But the real trick came when we combined the two modified nucleosides. Of course, they're on different nucleosides, so we could do that, put 5-methylcytosine and pseudouridine into the cells. Now we got very, very good viability, above 80%, and very robust protein expression. So we started to use this technology, which we called modified mRNA, or modRNA for short, with these modified nucleosides, and characterized its properties. It had, really nice, I'll say, drug-like properties. So here on the left-hand side of the screen, we're again just transfecting mRNA for GFP, and you can see in a dose dependent way that the more mRNA we transfected, the more cells got transfected and the higher the signal. So from 10 nanograms to 1,000 nanograms, you've got a really blasting signal of almost 100% of the cells expressing the GFP, and that's quantified on the right here. But there's one thing that was certain, and that's that, mRNA is a relatively labile molecule. And by the way, this is why I think many researchers, although I suspected many researchers tried to use mRNA in the, decades since, the early 1960s, but many were afraid to use it because it's very a labile molecule. It's very easily degraded by RNases, which are all over our pipettes and all over our bench and all over our hands. And that was kind of the belief, that people didn't want to use it because it was too labile. But actually, I think the real reason is because it's highly immunogenic when you reintroduce it into cells. Anyhow, when you, one thing that is true though, it does have a relatively short half-life even in cells. So when you transfect it, this is just with a GFP time on the x-axis here, you can see that, post-transfection at, 0 hours, by 12 hours, you had sort of maxed out expression, and it returned to baseline a couple of days later. And that was with a relatively long-lived GFP protein. We also did it with a shorter-lived GFP protein, and that time window was shortened, of course, because there's two things at play here. There's the half-life of the mRNA and also the half-life of the protein that you're making. But we found that we could actually, and this was really only true of the modified RNA, which was non-immunogenic compared to unmodified RNA, we could actually transfect on day 1 and then go back again and transfect on day 2, day 3, and on and on. And so this is an experiment. We transfected 10 days consecutively in these human fibroblasts on a plate. So the first thing you can appreciate is that the plate is full of fibroblasts. There's a monolayer here. So they're not dead. They're really happy. And in this case, we were using a nuclear localized GFP, and you could see that here. I just overlaid the GFP channel with the bright field. Another nice thing, you could transfect more than one mRNA at the same time and get both expressed. So here we had an mRNA for M. cherry, another one for GFP, transfected into human keratinocytes, and all the cells express both of the mRNAs, both of the proteins. We transfected up to 7 at a time, and all were expressed. I know Drew Weissman has told me of experiments of 25 at a time. So really the number of mRNAs that can be transfected, it's all a matter of getting them into the delivery vehicle, and you could make, I used to call them sandwiches of all kinds of different ingredients of mRNA. And if the sandwich goes in, if the cell eats the sandwich, all of the ingredients go in. So actually an experiment that I tried to convince multiple postdocs and students to do, which nobody did. Nobody would take me up on the challenge, which was to take a cell type that totally didn't have an entire signal transduction pathway, from cell surface molecules to cytoplasmic mediators to transcription factors, and express the whole cascade of signaling transduction of a signaling transduction pathway in a cell type that didn't have any of these elements. And I just thought that would be a really cool way to sort of demonstrate the power of the technology. But like I said, nobody bit at that one. So we went back to our task at hand, which was moving cell fate around. There's many other experiments which I don't show you characterizing the technology. But the first thing we did was we moved fibroblast cell fate around. We just did a 1-factor cell fate translation or transformation. So we express sort of a master myogenic transcription factor, MyoD. And these are from the very early Weintraub experiments showing that MyoD could convert the fate of fibroblasts to multinucleate myofibers, which I show here on the slide on the right, multinucleate. They were contractile. They would actually pull themselves off the dish. We, of course, went back and did RNA reprogramming to pluripotency. And really the remarkable thing, and this was not expected, but so I'm showing you an image here of pluripotency marker immunostaining of a plate of cells that we reprogram versus the Yamanaka reprogramming. And sort of the brown dots are the cells that are expressing this TRA160 marker that are reprogrammed. So you can see Yamanaka reprogramming was relatively inefficacious. Mind you, this is not a terribly ineffective experiment. There's plenty of pluripotent colonies there. But if you look at the plate of our cells, it was pretty much the whole plate was converted. And this is with the original 4-factor Yamanaka. If we added Lin28 to that, we literally turned the plate brown. You couldn't see anything that wasn't reprogrammed. And so the effectiveness was, up to 40-fold more effective than retroviral reprogramming. And then we did an experiment, which I thought was really cool, which got largely ignored in our paper when we published it. But, at the time, the elephant in the room for using iPS cells for therapeutic purpose, disease modeling and/or autologous cell and tissue transplantation, was that the methodologies for turning pluripotent cells into a differentiated cell type were largely driven by these by modulating the extracellular milieu of iPS cells to coax their fate towards a given lineage. But I thought, well, we could maybe just, introduce factors that would, instead of program in a forward direction cells to a certain fate. So we did this experiment. We first reprogrammed to pluripotency with the modified RNA, and then we took those cells. So those were fibroblasts, and then we added a MyoD RNA. First, we let them just sort of grow out a bit, and then we took MyoD and we turned them into multinucleate myofibers. And I actually thought that this was going to be the thing that people really sort of latched on to. But I honestly don't think I've seen anybody do such experiments since. And still, the way that people derive their sort of differentiated cell types from a progenitor or a pluripotent cell is through modulation of the extracellular milieu with cytokines and growth factors and the like. So we published this paper in 2010. We got the cover of Cell Stem Cell, which was cool, and it made a lot of. It was very celebrated in both the lay and scientific press, which was cool. But what I found a little bit surprising was I was getting, calls from, pharma executives on a relatively regular basis saying, Dr. Rossi, we saw your technology for making iPS cells. We'd really like to license that because we have an iPS cell sort of program. And of course, all of pharma was doing iPS cell programs at the time. Remember, this is only 3 years or 3 and a bit years post Yamanaka's original discovery. But what I didn't hear any of those pharma guys say was, wow, you can really make any protein in cells. Wouldn't that be cool? Thinking about a therapeutic. Found that really surprising. But that's what I was thinking about. So I thought, well, what experiments would we need to do to convince ourselves that, modified mRNA might be used translation as a therapeutic? So we had done all our work in tissue culture cells, human cells, but in a dish at the time. So of course we had to take it into animals. And so we did an experiment where we made a modified mRNA for luciferase. And this is literally, I don't think this is the first experiment, but the first experiment was as successful as this. So we took it downstairs, a bunch of lab knuckleheads. We didn't know anything about delivery. We just did it in the same sort of lipoplex that we had been transfecting cells in the tissue culture dish. And we took down varying doses of luciferase mRNA, modified mRNA, injected in the thigh muscle of mice that had been dosed with luciferin, the substrate, and then put them in the CCD chamber. And lo and behold, we were getting really robust expression. And it's actually a 9-hour time point, but even as soon as 2-3 hours post transfection, we could see light emitting from the thigh muscle of these mice. And the cool thing is, which maybe you can appreciate from the x-axis at the top here, is relatively dose dependent. And again, this was very primitive cell delivery technology we were using. So this is the work of Wataru Obina and Lior Zanghi. And I should have said that the iPS cell work was largely driven by Luigi Warren in the lab. Luciferase is not a therapeutic protein, so we wanted to have some evidence that we could do something translationally. So we made a modified mRNA for human erythropoietin, and human EPO acts on the mouse erythropoietin receptor to drive red cell production. We knew this, but we made the human EPO because it differs from the mouse EPO in an epitope that we could pick up with an ELISA assay. So you could see that in this experiment here, that we dosed, again, we put the mRNA for EPO into the thigh muscle of the mice, and in a dose-dependent manner, we could pull up erythropoietin floating around the blood plasma levels in these animals. And when we did their red cell counts, we saw a dose-dependent increase in red cells and hematocrit and hematoglobin. This is the work of Pankaj Mandal and Morag Stewart in the lab. We also did a study that was published a couple of years later, and this work of Lior Zanghi, where he was very interested in the heart and cardiac infarction and the problems that emerge from infarction. So you get an infarction in your heart, and then you get a fibrotic repair process, which is leads to diminished heart function. So we thought, well, there had been some large body of literature suggesting that, well, if VEGF, vascular endothelial growth factor, might be able to be used to stimulate neovasculogenesis in the damaged heart, and that might reduce fibrosis during recovery from infarct. So we went into the murine heart. If you go in with DNA, and this was a reviewer question, so a DNA, and here we're transfecting a CRE, either CRE DNA or a CRE modified mRNA, and we're putting it into LOX/LAC-Z mice. So only if CRE is expressed does it flux out the stop codon, and LAC-Z is expressed. So wherever you see blue, that means that the mRNA was expressed. And you can see that the mRNA was really robustly, and again, dose-dependent expression in the murine heart. What I find interesting about this is that we did a single needle injection, and you can sort of see it here. I think the needle track is somewhere around here. Not totally sure about that, but it's sort of in the middle of the of this wave of expression, suggesting that it was likely there. But more than just the needle track of cells got transfected. Really, a pretty large swathe of the murine heart was transfected. You really see it with this laxine staining here. That was a single injection. This program eventually got picked up by AstraZeneca, actually, and taken through phase 2. But they've ended the program this past year. But you know, with the idea of for infarct patients coming in and getting a dose of modified mRNA for VEGF into their newly infarcted heart for regeneration. But as I said, unfortunately, that program was discontinued. But our original paper was in Nature Biotech in 2013, and this just showed that. So I'm sorry, I started with VEGF. Here's the VEGF experiment here. So if you put a modified mRNA for VEGF. If you don't do it, this is the type of vasculogenesis you get on an infarct heart. You don't get much neovasculogenesis, which are these orange, these stained with these orange squiggles, I guess. Whereas if you put it in VEGF or modified RNA, VEGF modified RNA, you get really good vasculogenesis upon repair. And DNA, you got some, but not quite as good. And this is 7 days post-infarct, and it's microfill staining that we're looking at there. A modified mRNA had some really good characteristics. So I put together a pitch deck, and I went out to venture capitalists to try to raise some money, and actually colleagues first. Nice properties. It's non-toxic. These are naturally occurring nucleosides, non-immunogenic. That's what makes it work. It's dose titratable. That's good for a drug. You could control temporally whether or not you wanted a short burst of protein expression or sustained by either doing a single or multiple deliveries. And really the big thing here is the versatility. Really, any open reading frame that you knew the sequence of could be turned into a modified mRNA to express that protein. In my lab, within the first sort of 6 months of sort of working with this technology, we had over 100 mRNAs, modified mRNAs that we're using routinely in the lab. All the lab people used it in their projects. Timeline for drug development would be fast, I imagine. When we wanted to make a new modified mRNA, if we had a cDNA or we'd ordered the cDNA or clone the cDNA and put it into a vector for reverse transcriptase with these modified nucleosides and purify it, we could do that in a week and a half. So very, very rapid to make a new testable modified mRNA, and you can imagine the efficacy being read out in 1 to 3 months, whatever your model happened to be, much, much faster than protein therapeutics. And so I imagine sort of disrupting the protein therapeutic sort of paradigm. So there's over 100 different FDA approved recombinant proteins, but now I imagine you could do this at much better cost of goods, because now you could have the patients make their own protein, and it would be properly post-translationally modified because it would be made by the patient's own cells. And again, the diversity of things that you could do, you could make secreted factors using sort of a depot strategy, as we did with that mRNA for EPO, or localized intracellular proteins or nuclear proteins, stuff that's normally out of bounds for recombinant protein therapy, which largely works in the extracellular milieu. So after raising some funding from an angel, Tim Springer, who was a colleague who founded Millennium, I recruited a couple of co-founders, Bob Langer at MIT and Ken Shen, who is the only MD, Ph.D. or MD, and I'm a PhD. Bob's an engineer. I wanted to make a therapeutics company, so wanted to have somebody that knew something about medicine. We raised $2.5 million, small raise in 2010. These were original labs over here on First Ave in Cambridge, Mass. So we, as I said, raised $2.5 million. Before going public, Moderna raised over $2.5 billion. So add 3 zeros to that. That's how much money was raised. Went public in 2008, raised a record amount of money at the time, over $600 million. Nice ticker mRNA. This is an old slide. I don't know what the market cap is today, but somewhere in the 70-ish billion range. Again, this is old, but currently I think there's about 2,500 employees, lots of job postings. It's a multinational company now. Importantly, Moderna used some of these billions raised to build a manufacturing facility prior to COVID happening in Norwood, Mass. And that's actually what accelerated the development of the COVID vaccine when the time came, because we already had the manufacturing capability of doing it. And again, this is all, many programs in clinical development in lots of different areas. As I said, vaccines, cancer vaccines, secreted factors, intracellular therapeutics, immuno-oncology. I think oncology is going to be a big, big deal for mRNA science. Again, this is old. I don't know what the pipeline looks like today. This was probably a few months ago. Various different programs at various different stages. It's just another slide of that. There's, I know this is in phase 3. The RSV program is also in phase 3. Yeah, so here, this is a little bit old because this is now over here. But of course, the one that everybody knows about is this COVID-19 vaccine, which was emerged in the face of SARS-CoV-2, which got declared a global pandemic in March of 2020. Moderna started working on this after the sequence to SARS-CoV-2 was published by the Shanghai Consortium in January of 2020. And 45 days post learning the sequence, they shipped a patient-ready sample of the mRNA vaccine against spike protein. And this is the clinical data, which I'm sure many of you have seen from the original randomized phase 3 trial, where those that didn't get the vaccine, they got placebo, they continued to get this is cumulative event rate. So they continued to get COVID-19, whereas those that got the mRNA vaccine were largely protected, 95% against infection and 100% against hospitalization and death. So we know all about that now. The original strain was 95% effective. Delta came around. This is a great pathogen, just a real a fast evolver. Well, with so many replications and so much opportunity for natural selection to occur, it's done that very effectively in a really short period of time. So Delta, the original strains were only 70 to 80% effective at. If you boosted, okay, you got up to 90. The Omicron strains, the original Omicron strain, only 20 to 30% effective, and even upon boost, 70, 75% effective. And this is data from prior to the latest versions of Omicron. So Omicron, as you know, has 54 mutations. Luckily for us, it's a less pathogenic, less virulent virus, but it's a super highly contagious virus. So it's raging. I literally just got a text from a friend whom I was supposed to get together with tomorrow in New York. And he's like, oh man, I think I got the COVID. So I won't be seeing my friend Michael tomorrow. But lucky for us, the bivalent boosters have been emergency authorized approved. So Omicron plus the original SARS-CoV-2, and that was in August of this year. So it's available. But we can also, one thing we can be sure of, we're only halfway through the Greek alphabet, and Phi, Rho, Sigma, all the way to Omega are likely to emerge. So we have to stay vigilant. I give this talk sometimes to lay audience. So just a reminder, this is not a lay audience. So bone marrow transplantation procedures, there's about 50 to 55,000 performed yearly globally, and this is just sort of a heat map of where bone marrow transplant or mobilized peripheral blood transplants are performed yearly, and it's a great therapy. It's life-saving, or can be life-saving, for a number of really deadly diseases, leukemias, lymphomas, bone marrow failure syndromes, sickle cell, thalassemia, MDS, and others. But there's no question that it remains a high-risk procedure. So that's bespoken by these numbers here. So the 1-year survival rate for HLA matched siblings is only 70% and 55% for unrelated donors. These are a little bit older numbers. I think these numbers are from 2013. What kills patients largely is the relapse, depending upon what they're being transplanted for. But, opportunistic infection, graft versus host, is common, really unwanted side effects of bone marrow transplant. But it is life-saving for many. So first transplants were done, over 60 years ago. It's curative. Over 1 million patients have actually been transplanted now. And I actually love this picture here. I don't know if you can, the top left, I don't know if you can see my laser. But this is the oldest surviving bone marrow transplant patient, Nancy McLean. This is her transplant doc here on the left. Robert Kyle, this is 52 years post original transplant. And what I think is the most interesting part, well, not the most interesting, but I actually think the photographer did an injustice by only sort of getting half of this person in profile. But if you look closely, you can actually sort of see that this woman looks an awful lot like this woman. It's because they're identical twins, and she was actually the bone marrow donor, and her twin had bone marrow failure syndrome. But that was in the days before HLA matching, so you could really only transplant twins. But then we learned about HLA matching, so you get transplants from a donor such as Petra Polker here on the right, with her the person who received her bone marrow, this young woman, Jocelyn Miller, on the left, who got cured of sickle cell, very severe sickle cell disease, and they were a good HLA match. But of course, as I said, a very severe, it's a really last ditch a procedure that you'll be prescribed because of all of this untoward morbidity and mortality. So we were thinking about this, we and many others around the planet, and this is how the procedure usually happens. You have a donor, you collect their bone marrow or mobilize peripheral blood, you process it, you maybe freeze it, you unfreeze it, and here's your donor. They need to be sort of prepped for transplant, so conditioned. So with either high dose chemotherapy or radiation, does two things: clears out their old marrow, possibly hopefully does a number on whatever cancer they happen to be plagued with, blood cancer, for example, and then the donor graft is infused. So we imagine that, and there are some people that are not good mobilizers. You could, not everybody mobilizes well. It could be a good donor, but a poor mobilizer. One thing is true: stem cells don't expand very well. You take them ex vivo and they differentiate. And honestly, the single biggest factor for transplant success is the number of stem cells transplanted. But this is really a big deal here, conditioning. If you didn't have to give non-discriminant sort of conditioning to a full patient, you could really just target the bone marrow or stem cell compartment. It would be preferred. And you can imagine making transplant safer and not only using it for standard of care settings, but curative therapy for many other things for which there's good case studies suggesting that bone marrow transplant can be used, for example, to treat people with autoimmunity, for example. So we did some experiments in the lab where we took a relatively stem cell specific antibody targeting c-KIT, CD117, also known as. It's expressed on blood-forming stem cells and their most proximal multipotent and oligopotent progeny, but not on differentiated blood cells. It's also expressed in a few other cell types of the body. And we hooked up we got a good antibody for that and put an immunotoxin on it and conditioned mice with this, hoping just to ablate the stem cell compartment, stem progenitor cell compartment, so we could transplant. And indeed, that turned out to be true. So if you don't condition the animals and you transplant an unconditioned animal, you don't get very good engraftment. So that's in the gray bar here, and the level of chimerism, the level of engraftment is here on the y-axis. And I'm waving this as year post transplant. Whereas if you condition with this immunotoxin conjugated antibody, you could really get really robust chimerism. So Anishka Chekowitz in the lab, who's at Stanford now running her own lab, published that paper in 2019. We did it collaboratively with Raul Patterjee, who's a co-first author on this paper, published the original paper where they did the same approach targeting CD45, which is pan-hematopoietic, but again, with the intent of transplantation. So with that data, I co-founded Magenta Therapeutics along with my colleague at Harvard, David Skadden. This was the early team. This is, again, I'm sorry, this deck is quite old, but this is what their sort of pipeline looks like. But public went public in 2018, the ticker of MGTA, but a couple of programs in phase 1 and phase 2. The conditioning programs are what I'm most excited about, that I think is going to be the most impactful. This is a mobilization program. We had a better mobilizer. This is the sort of c-KIT targeted conditioning that Anishka and my lab led the charge on. And this is other programs which are preclinical. And so one last story. So gene editing, and again, this starts again with an unlikely source, and it's this Timothy Brown, also known as the Berlin patient. Many of you in the audience, I'm sure, know. He was twice unlucky and once very lucky. So he was unlucky in that he was HIV positive, but he was really unlucky when he was diagnosed with acute myeloid leukemia. But he got lucky in that he was sort of not one of the first wave of HIV patients, but a later wave. And it had already been known that patients with people with a certain deletion of a gene called CCR5, which is a co-receptor for HIV-1 strains of HIV, when there was a naturally occurring mutation called Delta 32. And so it's a small deletion inside the CCR5 gene. But when you were homozygous for this CCR5, which some people in the Northern Hemisphere are, they were highly resistant to HIV infection, HIV-1 strains of HIV. And so this was known. And when he needed his transplant, Timothy Brown, his doctors found somebody who was HLA matched and from a Delta 32 donor. So he became the first person cured of HIV. There's now actually been a second patient, the London patient. So this is potentially a way to cure people of HIV. Antiretrovirals are great, of course, but if we could cure people of their disease, might we do that? So when CRISPR was first published, when it made the jump out of bacterial and bacteriophage biology in 2013, we read this paper and we thought, well, let's apply CRISPR to blood-forming stem cells and let's target CCR5 and to show that we could make a delta allele with human HSCs that could potentially be used as a therapeutic for patients such as Timothy Brown. So Pankaj Mandal in my lab led this study. We actually made a dual guide strategy. So to make a delta allele, we made lots of different guides. We used lots of different, well, a handful of different human donors. The nice thing about HSCs, you can clonally screen them. You can make them form colonies and examine the clones to see that we had pretty good monoallelic inactivation and pretty high percentage of biallelic inactivation. When CRISPR first made the jump into mammalian cells, the efficacy was 1% to 2%, 3%. It was terrible. We were getting, 20, 25%. So using electroporation of CRISPR into blood cells, we were getting a lot of cell death from electroporation. But if we got CRISPR in there, we could really get it to work with some good quality guides. Those cells that were CRISPR treated, we could transplant them into NOD-SCID mice, get human chimerism, multi-lineage, B and T cells, or I'm sorry, B and myeloid cells here, but T cells as well. And we could take the bone marrow, the spleen out of those animals and show that this delta allele that we made was engrafting into the mice. And actually, which I don't show you the data, it was very, very highly efficacious and very, very highly specific. We did a very, very deep off-target analysis, and really, if you make a good guide, you could get really specific cutting. So what we envisioned was taking HSCs out of a patient that happens to be HIV positive. They're positive for CCR5 using CRISPR Cas9 to make a delta CCR5 allele and transplant back in. And wouldn't it be nice if you could transplant back in a non-myeloablative way, a la Magenta? And actually, what's kind of cool is the HSCs are being used to treat patients with CRISPR, not for CCR5, but for things like sickle cell and thalassemia and other diseases. But the neat thing is, and I'm kind of quite happy about this, the Cas9 is being expressed almost exclusively in all these studies by modified mRNA. So kind of we had a little bit of a role in that, and hopefully I should put in a Magenta sign or a c-kit conditioning here. I got to put that into that slide. We also got the cover of Cell Stem Cell. Again, like I said, it was very precise editing. So that's what was depicted in the picture there. And that was published in 2014, November. And then 4 days later, we launched Intellia Therapeutics with a bunch of CRISPR pioneers. Jennifer Doudna, won the Nobel Prize, but Luciano Marafini, Rudolf Barangu, and Eric Sonheimer are early CRISPR pioneers when they were, when CRISPR was still a bacteria, fighting off bacteriophage issue. And this is, again, an old slide, but it's in the clinic in phase 1, moving to phase 2 for a number of programs. Actually, this is exciting. In vivo CRISPR, so putting the mRNA for Cas9 and the guides into lipid nanoparticles, putting them into people, getting it to go to the liver and target these targets. And that data, actually, early data, clinical data looks very good. But you can also take the cells out, for example, as we did with hematopoietic stem cells, and introduce CRISPR-Cas9 and then put the cells back into people either for, AML here, sickle cell, or CAR T therapies are being engineered with CRISPR and mRNA. This is I retired from academia in 2018, but this is what my lab looked like at the time. So many people to thank who did all the work, many collaborators, funding sources, and I'll stop there and take any questions if you have them.
All right, that was really a tour de force. Thanks so much, Derrick. I know you wanted to be here, but we really appreciate you sharing so much fundamental science with us. As you can see from Derrick's talk, when we worked together, it was 17 and a half years ago now, but who's remembering, in Irv Weissman's lab, we were working on stem cell aging, and you can see Derrick's continuous deep dives into science are completely based on his incredible inquisitiveness. I'm just wondering about any questions in the audience.
I'm good for time, Katrina.
You're good for time?
You don't have to rush. I've got a dinner reservation, but I'd be able to make it still.
Yeah, I had a question. I saw Beam Therapeutics on your slide, Derrick. And so I was interested in prime editing, so using APOBEC base editing and also ADAR-mediated base editing. Have you looked into those strategies? You use standard Cas9, CRISPR, or not so standard, your modified RNA version?
No, I have not, but you should, because you're focused on ADAR and base editing. No, but for sure others are. I think it's a great avenue to pursue. Base editing is a smart idea. Obviously, the beauty of base editing is getting away from double-strand DNA breaks. You know, the problem I always had, and I remember early strategies where people were talking about, for example, targeting viruses in cells with CRISPR. It's a terrible idea where you have multiple different, and this is the whole danger of off-target, is as soon as you make a double-strand break, if you have another double-strand break somewhere else in the genome, you're going to guarantee at some frequency those two double-strand breaks are going to align. You're going to get translocation. So if you don't have to go for double-strand break and you could rather base edit using various strategies, it's a better strategy. You know, you have to imagine there is a beauty to cutting the DNA, let's say for making delta alleles, like as the case for possibly for HIV prevention, CCR5. But I think that, and if you could do it in, with really highly specific guides that don't cut elsewhere in the genome, then there's a path forward. And there is a path forward because the FDA is very, very cognizant of off-target. That's like one of the biggest things that one gets grilled on when you take a therapy, a CRISPR therapy into the clinic. So you really have to convincingly show that your level of off-target is very low. By the way, gene therapy is not considered, for integrating gene therapy, those are double-strand breaks to get those vectors into the genome. And we know sometimes the problem is that the early generation of lentiviruses, activated oncogenes, and that was the problem. But the fact of the matter is there's double-strand cuts to get them into the genome. So we can cut the genome safely and efficiently. There is, of course, it's a numbers game. At some point, the numbers, even if it's 1 in a trillion, you're going to hit that number and have that adverse event. So it's a safety versus efficacy. You must weight that. But certainly base editing without the DNA cutting, if you can get the efficacy up to very high levels, like, I showed you some data, and that's old data. That's from like 2013. Very, very high levels of efficacy now, into the 80, 90, 100% range with CRISPR cutting now. You really need efficacy too. And I haven't looked at the base editors of late, but they have been less effective, at least. But I'm not up on the field, so it could be that the efficacy is going up on those as well.
Yeah, so I think Rob Signer has a question, but just before we do that, I remember in 2010 when you developed the RNA and pluripotent stem cell technology, RIPA, and you were in Time magazine. You were so excited about that and being honored, and you've clearly personified that Ganbatsei, never ever ever give up spirit that you hear about in Japan. What made you keep going? I remember you were having trouble getting the company financed. You said, I know this is a great idea and nobody's funding it. How did you solve that issue? What made you keep going?
Well, I mean, it's true of any scientist sitting in the room when you know something to be, and we only know as far as our experimental data can confirm. So hopefully we know when we feel really positive about a data or a platform or a technology or something, you just, you find a way because, and you can convince others that there's a way forward. So, yeah, moreover, we have a lot of training as scientists in defeat and hitting the wall. So it's
Too
not,
much.
that's one thing we're trained at, is like really getting punched around quite a bit to get our papers published and get our grants written, and even our experiments to work. Honestly, if you have a experimental success rate of 20%, you're doing pretty damn good. So we're accustomed to failure. You just got to keep going. It's true of every single person in the room there. Scientists have that in them.
That be an MMA fighter. I love it. So Rob has a question. Rob Signer.
Hi, Derrick. Thanks for a wonderful talk. My question's about delivering mRNAs to, hematopoietic or other somatic stem cells, and the issue with them having really low translational output. And if this is creating a roadblock to actually getting those mRNAs to express protein within those stem cells.
Yeah, it's a good point. So, HSCs, hematopoietic stem cells, a pretty dormant cell, very, it's all nucleus. It's almost no cytoplasm. It doesn't need a cytoplasm. It's not making a lot of protein synthesis. If it's resting in G0, it's not going through the cell cycle. It's taking a little pause there. So, if you were doing it ex vivo, and as soon as you take HSCs ex vivo, you almost activate them by default. So they go into cycle any type of stimulus. And you can actually, the cool thing about HSCs, when you take them ex vivo, they're all cell synchronized. They're G0 and then they go into G1. Actually, they have to clean up a bunch of DNA damage first. We published on that. They come out of G0 and first thing they see is, oh, our genome's full of damage. We got to clean all that up. But then they enter G1 more or less synchronously, and you can time S phase actually. So you can actually, there's an advantage if you want to take advantage of HSCs when you first pull them out. They're cell synchronized and offers advantages. In vivo, however, you've got a really great point. I mean, the bigger problem with in vivo, and you mentioned it, the delivery, getting a cell type specific delivery vehicle is, I think, going to be the next big frontier in delivery science for mRNAs. The great news is lots of people are working on it. I mean, even if you look at the history of lipid nanoparticles, when they were first developed for siRNAs and the like, the efficacy was orders of magnitude less good than it is now. Moreover, they were orders of magnitude more toxic. So they're getting more and more effective, less and less toxic. But I think the next frontier is more and more cell type specific vehicles. It doesn't have to necessarily be lipid nanoparticles, be nanoparticle of some sort, or viral, possibly viral delivery or viral shell capsid in some way, shape, or form, or other technologies that aren't yet developed or being developed, and then getting into a cell type that is, I think your point, which was a good one, a relatively low protein turnover, and let's say you need to make Cas9. Well, there's low protein expression, but there is protein expression even in dormant cells. But you could always give a stimulation to a patient, let's say, to push these cells into cycle. That's possible to do, to up the protein translation if you needed to have something expressed. But I think it's a really good point, because it's true. It's a really dormant G0 population, and the bigger point of targeting those cells in a specific way, I think, is really a great frontier, and something people are working on, and more people will work on.
Well, thank you so much, Derrick. As you can see, Derrick has gone through the 3 phases of the truth, just like Liz Blackburn did with her discovery of telomerase. First, it's ridiculed, then it's vehemently opposed, and then it's accepted as self-evident. And that's usually how you win a Nobel Prize. So we're rooting for you, Derrick. But
I think I am a little bit surprised that Ketty and Drew didn't win it this past year. I mean, their fundamental discovery of the modified nucleosides are really, we took it, we put it into, made the translational leap, we and many others, but that's the fundamental discovery. So I think they'll be recognized. I was a little bit surprised it didn't happen this year.
Very humble as usual. So you can see Derrick's a true blue Canadian with that humility. But also, I think it just caps off the day perfectly. So you heard the fundamental discovery of longevity, what fuels longevity, from Elizabeth Blackburn this morning. We ended the day for the reason most of us have longevity thanks to your technology, Derrick, with Moderna. We wouldn't be here in person if it weren't for you, Derrick, persevering, so thank you. And clearly there are many more things to do to treat other diseases, but certainly you allowed us to get through the pandemic. And now, instead of being holograms, we're actually here in living person, and hopefully we'll have you here again soon. I'm just going to wrap up the day very quickly. Thank you again, Derrick. That was an incredible scientific tour. See you soon.
Ciao, ciao.
Yeah, thanks.